Hello, everyone, and welcome to the Sexton Biotechnologies webinar, BioDefined Media for Next Generation Therapies. My name is Dusty Howe, and I am the Marketing Manager at Sexton Biotechnologies. Today's webinar will include presentations from our two speakers, Aoife Duffy and Sean Werner, followed by a brief Q&A session. Please feel free to submit questions in the comment section, and we will get to as many of those as we possibly can. We also wanted to take this opportunity to announce that our partner in this webinar, HiTech Health, is announcing their conference, Cell and Gene at the Abbey, taking place in Galway, Ireland, at the Glenlow Abbey Hotel on November 10th through the 12th. During the Q&A, we will provide a slide with more information about how you can participate. Our first speaker today is Aoife Duffy, Cell and Gene Therapy Operations Manager for HiTech Health. Aoife has almost 20 years experience in ATMPs, GMP manufacturing of ATMPs, quality control, and regulatory affairs. Aoife played an essential role in getting the regulatory license for the first approved ATMP manufacturing facility in Ireland to produce ATMPs for clinical trials, where she led the technical transfer, manufacturing, and regulatory affairs. With HiTech Health, Aoife is leading the cell and gene therapy operations, where HiTech Health have a process development laboratory operating under GMP to perform key projects including, but not limited to, cryopreservation studies, bioreactor testing, process optimization, analytical and release assay development, and bespoke training packages. In addition, HiTech Health are working with clients from across the world in cell gene therapy to provide QP services as well as a regulatory support and project management. Go ahead, Aoife.
Thanks, Dusty, for the introduction. My name is Aoife Duffy, and I am the Cell and Gene Therapy Operations Manager at HiTech Health in Ireland. I am delighted to be here today to present on BioDefined media for next generation therapies. On this slide is an overview of the current services provided by HiTech Health. We provide both advisory services and also process development services at our laboratory based in Galway. On the advisory side, we provide services such as qualified persons for cell and gene therapy products, importation of products, independent auditing of CDMOs or suppliers, development of QMS systems, and we support in developing GMP compliance into your process. At our process development lab in Galway, we work with clients developing quality control and analytical assays, scale-up development, process improvements or changes to filed processes, cryopreservation studies, shipping studies, bioreactor assessment, and we also do bespoke training packages for cell and gene therapy clients. So as we all know, the cell and gene therapy space is an ever-expanding industry, which has a growing demand for supply for hundreds of disease indications. Traditionally, processes were developed in R&D laboratories using R&D grade materials and small scale with often a lot of open aseptic processing. Once the product gets to first phase approval, it's difficult to put the time and money into revalidating using more suitable raw materials, as often the drive to get to the clinic outweighs the revalidation to more suitable materials. And with that brings challenges, such as limited raw material sources ongoing for phase 2 and phase 3. Incorporating key raw materials suitable for large-scale manufacturing with no supply issues is key for all future products, as well as current processes that may need revalidating. So what are the common growth supplements out there for cell culture right now? We have fetal bovine serum, human platelet lysate, AB serum, and chemically defined serum-free media. And as we know, there are multiple challenges with FBS. Firstly, there's a high demand and a low supply. Large companies take the bulk of the supply from Australia and New Zealand, which are the preferred sources, and leave it difficult for smaller companies in phase 1 trials to get access to it. And as we know, for phase 3 trials, you do need to be required to complete this. It's a fragile market in that if there's any outbreak of BSE, it could be detrimental to the whole supply chain for FBS. And most importantly, from a regulatory standpoint, we want to move away from animal-derived source products in our manufacturing processes. So what are the other options outside of FBS? We have chemically defined media, but there is a limited utility of this to date, and the field is less eager to make the switch. There is also AB serum, but this is not optimal for culture of a lot of cell types, and there is also a supply concern. We have human platelet lysate, which is widely used in the field, and there is extensive literature using HPL for cell therapy products, with multiple companies and organizations developing various options for HPL. With that, there are challenges in the variability of HPL, and there also needs to be a minimum set of criteria for the HPL production and characterization. So with blood-derived products, there are regulatory challenges also and guidelines that we must adhere to. In the recent European Pharmacopoeia, it does state that the implementation of a pathogen reduction procedure in the manufacturing process is suggested, and there is also the option for low donor pools of HPL also. And as per the guidelines on ATMPs, a risk-based approach is needed for key raw materials in manufacturing, and understanding the impact of these on the manufacturing processes, as well as their intrinsic properties, which could affect the final product, is essential. So how can suppliers help in standardizing the HPL? Well, standardization of policies and procedures for HPL manufacturing processes assure production of a safe and consistent HPL product with lot-to-lot consistency. Manufacturing processes should be CGMP compliant with quality oversight. This should include in the environment, equipment, personnel, and manufacturing supplies, as well as monitoring, validation, training, and qualification. Traceability back to the blood donations and testing sites are also essential as part of the filings for CGT products, and with traceability back to the test kits required for filings also. And don't forget the qualification programs in-house. Qualifying reagent suitability is essential as part of a manufacturer's responsibility. Grow promotion against previous lots is highly advised to show that the product performs as expected. So what are the options with HPL at the moment? We have the small donor pool HPL. The Pharmacopoeia allows you to use small donor pools of 16 donors or less. However, the issues with the small donor pool is that there's large batch-to-batch variation, as well as the high cost due to the low volume products. On average, the small donor pools only generate around 2.5 litres, which wouldn't be enough to complete a trial. In addition, there's challenges with testing requirements on the HPL, as there can be different regulations between countries, such as between the EU and the US. HDLV testing is required by Ireland and France, but it's not required in some other EU countries. But we have to remember that the HPL needs to be tested to meet all the requirements across all countries. So the other option is large batch virally inactivated HPL. Obviously, the advantages of this are greater than the small donor pools in that we have greater batch sizes. It meets the regulatory requirements of viral inactivation as for the European Pharmacopoeia and also the guidelines in the ATMPs. There's less batch-to-batch variation due to the number of platelets used for the manufacturing, and it also reduces the cost from the manufacturing point of view as there's less rescreening required in incoming testing, where multiple batches previously would have been required. So at our process development laboratory in Galway, we only use the Sexton HPL for processing of our ASCs and our umbilical cord MSCs. And obviously the reason for why we're moving away from FBS is that we want to align more with regulatory requirements and also future manufacturing requirements for our processes. We use HPL at a concentration of 5%, and that's sufficient to give an excellent growth rates within our cultures of ASCs and cord MSCs. And the doubling times and population doublings range between 26 and 37 hours, which is comparable with yields and results that would have been obtained with FBS. And here I'm going to present some data that we have on our cultures, our human adipose-derived stromal cell cultures using the Sexton HPL. And here you can see from the morphology, the cells have the perfect MSC morphology, which are spindle-like and healthy cultures. In addition to the morphology, the doubling time for the ASCs was very good, with a range of 26 hours to 36 hours for a doubling. And in line with the doubling times, the cumulative population doublings over multiple passages were still in the exponential phase, and in line with what we observed from morphology and the doubling times. Finally, we demonstrated the cell surface characterization data met all the markers as required from the ISCT, and it demonstrated over 99% for positive markers and less than 0.5% for negative markers. In our hands at our facility in Galway, the HPL demonstrated great ability in the manufacturing of ASCs and umbilical cord MSCs, and the cells met all the criteria as expected. So for the future, being able to use the large batch virally inactivated HPL will be great for future manufacturing in our facility, and also is great as it meets all the regulatory requirements. With that, I'll hand you over to Sean to continue this discussion further. Thanks.
Thanks, Aoife. That was excellent. I'm now going to turn to our next speaker, Sean Werner. Sean is president of Sexton Biotechnologies, a manufacturer of processing and handling tools and reagents for the development and manufacture of cell and gene therapies. Sean received his PhD from Purdue University in biology, followed by postdoctoral positions at the Indiana University School of Medicine and Eli Lilly. Prior to his current role, he spent 11 years with Cook Medical and Cook Regentech, filling various roles in the global regulatory and general management functions supporting medical devices, autologous cell therapy, and single-use disposable development programs. In his 15 years working in the life sciences industry, he has guided preclinical and clinical testing and submission strategies leading to global commercialization of multiple medical device and bioprocessing tools. Take it away, Sean.
Thanks, Dusty. Thanks, Aoife, for giving that really insightful perspective on manufacturing requirements in the industry. We wanted to talk today about a concept we've been calling BioDefined media. Historically, there have really been two ways of looking at media composition. On the one hand, using a complex mixture of proteins derived from biological source material and just generally undefined biological supplements. On the other hand, you have exactly defined recipes using isolated proteins, chemicals, salts, sugars, and that obviously being a chemically defined media. But as we've been thinking about the undefined complex mixture of biologically sourced materials, we actually think there's a place in between the totally undefined mixture and a chemically defined mixture. Some materials that are used in supplementations actually have a pretty specific code that details what is present in those mixtures. And we think it's really useful to think about those in the context of those tissue sources' primary biological responsibility. We all know that one of the biggest challenges in cell and gene therapy manufacturing is overcoming the variability that's inherent in these processes. There's a large amount of variability in raw materials, in donor starting materials, in the culture processes generally. Everything from start to finish has a lot of variability that we only partially know how to control. And part of controlling that is really not controlling it, but getting parameters on the end, sort of boundary conditions that are acceptable. Qualify the processes to understand where you want to be within those limitations. Validate it so that you know at least you can contain that variability within those boundaries, and then continue to monitor it and make sure it hasn't shifted. In the end, what you end up with is a variable process, but you know at least where it's going and where it's going to end up within certain limits. So clearly, controlling as much variability as possible becomes critically important in developing a process that's going to end up with a reliable, robust manufacturing system to support commercially viable cell or gene therapy products. And that brings us back to the concept of biologically defined media. Ideally, you would be able to come up with a chemically defined media so that you eliminate that source of variability. But that can take years and may not even be possible in some of the processes that we're currently using. So if there's a way that you can limit the variability in the supplementation, there may be a midpoint between the sort of undefined serum-based product and a chemically defined product. We think that platelets are an ideal source of this kind of biodefined media supplementation. Platelets, though historically thought of as primarily involved in hemostasis and clotting, really have quite a robust immunomodulatory effect in the body, controlling the response at any given point during a response to an invasion or a wound. And in fact, in wound healing, they really seed the soil and guide the regenerative process. Based on the growing body of literature about the growth-promoting activity of platelets, human platelet lysate has emerged over the last few years as one of the most important supplements in cell therapy manufacturing. For Sexton, we developed Stimulate, our basic pooled human platelet lysate about 5 years ago, and brought that to market, commercialized it. It's available globally at the moment. As we went along and understood more and more about what the requirements were going to be globally for human-derived materials such as that, we developed Enliven PR, which is a pathogen-reduced version of Stimulate using a proprietary E-beam irradiation process. We went ahead and validated that process and found that that limited the impact of the performance on our platelet lysate compared to gamma or other methods. And then more recently, we've developed T-Liven PR, which is an irradiated product that is released against a T-cell specific growth promotion assay. And we really do think that platelet lysates should be thought of more as a product category instead of specific product, as in our hands, we've seen a varied performance and some very unique properties in each of these different products. During the development of Stimulate and Enliven PR, we recognized a pretty striking data set starting to emerge. As cell biologists, we're familiar with the concept of qualification of different lots of FBS or AB serum. Even within research groups, that's a common activity, really to make sure that you've got consistent data coming out the other side of your project. In cell therapy manufacturing, in the context of a GMP environment, that incoming qualification routine can be extremely expensive and may even be prohibitive and force you down the path of alternatives to FBS, for example. With Stimulate and Enliven, what we found was looking at total protein concentration, the variability was extremely low across a large number of lots, and performance was even less variable with our release assay. When compared to the variability within AB serum, our CVs were significantly reduced, clearly indicating a consistent product. That was a little bit surprising to us, given that in FBS, for example, the number of animals used to create a lot of FBS is sometimes in the thousands, compared to platelet lysates, which are commonly on the order of, say, 100 donors or around that. Since launching Stimulate several years ago, we've continued to do characterization studies on the product. And based on the reduced variability that we found, we undertook a proteomic analysis using a set panel of defined proteins and compared that to the proteins found in AB serum and in FBS across several different lots, both for Enliven and Stimulate, for AB serum and FBS. And one of the interesting things that we found is platelet lysate is definitely more than just serum. And if you go back to that BioDefined concept, where you think about serum and what is it and what's involved in that, and at some level, you can almost think of serum as the general transporters or the carriers, the logistics system for the body. It's clearly a media that the composition changes due to both extrinsic as well as intrinsic factors. Whereas platelets have that genetic code that defines what's generally within the platelet inclusions, what's in the platelets overall. So in the proteomic analysis that we undertook, there were clearly a lot more proteins, specifically in HPL, that were not in AB serum, and quite a number of them that were expressed significantly higher than AB serum. What we found nice about the array data was that we were able to target, in this case, about 640 proteins and look at them using the same technique across these different serum supplements. And that allowed us to get down to individual protein variability as opposed to the total protein content. And we found that it remained pretty consistent, that the variability in Enliven or Stimulate was quite a bit less than in AB serum. For example, in Enliven, we found that nearly a quarter of the proteins had CVs of less than 10% compared to AB serum, where that was only true in about 7% of the total proteins. And we found some pretty significant expression levels in some proteins that have a very important role in both immuno-oncology cell manufacturing as well as stem cell manufacturing. Proteomic arrays can be notoriously tricky, and it's always questionable about how much validity you should put on some of those data other than just using it as a guide. So we actually followed up and did ELISAs directly with specific growth factors that we were interested in, and it held up for that as well. So both in stimulate and enliven, looking at proteins such as EGF or VEGF, we found a pretty striking consistency across multiple lots. So what are the implications of a BioDefined media? What does it mean or matter to you in your process that we have higher levels of expression of both growth promoting and immunomodulatory proteins in platelet lysate compared to AB serum? One of the things we noticed as we were looking at the proteomic data was a relatively high expression of IL-7 compared to AB serum and some other immune cell promoting proteins. So we thought, in the context of cell therapy manufacturing, immuno-oncology applications, there's sort of a couple of different ways that people supplement expensive recombinant cytokines into their media. And so we were curious if, based on the composition of HPL, we might be able to reduce some of those cytokines or even eliminate them. So we undertook a study using the IL-15, IL-7 combination and compared the results in AB serum versus enliven. And what we found was that in our hands, you could eliminate the use of IL-7 altogether and actually titrate down IL-15 to a much lower level compared to the results in AB serum, where doing the same really didn't give you nearly as much cell expansion. Taking those results to the next level, looking at what the outcomes are then as you look at an overall manufacturing process, how does that apply to the variability in the overall system? Claire et al. Published in BioInsights last year some data looking at the reduction in variability across multiple donors when using stimulate or enliven in this case, compared to AB serum. And what they found was that, in fact, the platelet lysate enliven improved the performance across donors, evening out some of that variability. But the performance of cells in an in vitro assay or in culture is really not our objective. We're working on products that will cure patients of cancer or help reduce the immune system response to transplantation or help regenerate tissues. So we continued the analysis to look at what are the impacts on the final product if you use enliven to grow the cells. Watanabe and others published a paper looking at this, taking a CAR T product all the way through an in vivo model, both a liquid tumor model and a solid tumor model, and found that not only did the cells perform well in culture, they actually increased the survival in vivo in those mouse models. And that was really striking to us. And we think that it's a really important definition of how a BioDefined media can help make cell manufacturing processes even better.
Thank you, Sean. There was a lot of great information there in that presentation. So we've got a little bit of time left here. I'd like to get to some questions that we got during the presentation from some of our watchers out there. So first question that we got, what kind of regulatory experiences have you had with HPL? You want to take that?
Yeah, I can start on that. So I have previous experience in regulatory submissions using Stimulate, and for that we had to use the low donor pool Stimulate. With that we used the Sexton low donor pool Stimulate, but that had to be bespoke batches, basically small donor pools from up to 2.5 litres. And there were challenges with the regulators in that, because it's not terminally virally inactivated in any way, they wanted to identify right back to the source of each platelet procurement site the test kits down to the actual lot number of those kits that were used in the manufacture. And I suppose that was a challenge at the time of the regulatory submission, because you have to go back to each procurement site to find this data out and support that regulatory submission. And also, as I said, using the low donor pool, we could only get around 2.5 litres from every batch. And I suppose that limits your amount that you can use for production. And so multiple batches would have to be used over a clinical trial. And I suppose they were the challenges that we found with the low donor pools. However, they did get through regulatory submissions and they were successful. So we feel probably going forward to avoid those, using a virally inactivated HPL is more applicable for what we needed. And it was recommended by regulators in their comments that if we could use a virally inactivated source, it would be a better option for us. So obviously that's why we're looking at, the virally inactivated HPL right now.
Yeah, and I can add a little bit. Obviously we undertook the process of moving to Enliven basically to address the increasing restrictions or recommendations on safety for human-derived materials. And, our experience so far since we've launched that has been that it seems to meet the expectations. We've had a couple of clinical trials that we're aware of that have been approved within the European Union. We've taken information to Japan and have a certificate of suitability from PMDA, and we know we have use in the United States and FDA as well. So we feel pretty confident with where it is right now that it's we're keeping up with those more and more stringent requirements.
Fantastic. Cool. Second question we got, what formats does Sexton offer their HPL? Sean, I guess that's for you.
Yeah, so we have HPL. Stimulate is in bottles, 100, 500. We're expanding that to smaller volume bottles as well. Enliven comes in the same format. It's in bottles, 100, 500 mils. With the launch of T-Liven, we actually added bag packaging with different kinds of tubing that you can choose from, and a range of sizes from 50 mil to 500 mils, so that we're able to focus on small volume needs with early stage developers, but also scale with you as you're growing up and moving through the clinical process. We just sort of see that there's a more and more need for GMP compatible packaging for HPL or other ancillary materials as well. I don't know, Aoife, if you have anything on that experience to add.
Yeah, I mean, I think, Sean, what you're saying is correct, and having, different types of packaging available is important. From a manufacturing point of view, and from my experience, getting a bottle of 500 mils and having to aliquot that out, into 20 samples so you can use it to make up your media as you go along in the manufacturing process isn't ideal. You want smaller packaging that you can, weld on to your bags of media. And I think having, the different formulations is, from a manufacturing point of view, it reduces the aseptic risk if you can weld on a bag onto your system, rather than having to go through these large aseptic processes of aliquoting. I think it's ideal going forward.
Great. Next question we got. I noticed on the slide showing reduced cytokines that IL-2 was not mentioned. Do you have any data on that?
Yeah, thanks. So we ran IL-2 in the initial studies, and in our experience, the expansion was quite a bit less than in the studies with IL-15 or IL-15 and IL-7. So we're actually right now getting started on running the data with IL-2 by itself, just so that we can really understand what titrating that down looks like with our products.
All right, fantastic. And I've got one last question here. Why do you use E-beam versus gamma irradiation?
So when we started on the process of getting a virally inactivated or pathogen reduction technology, we of course started with gamma as one of the easiest things to do. And what we found was that in our hands, that really changed the performance of the product. We ended up with an HPL that just didn't meet our expectations in terms of cell expansion rate. So we looked at a large range of different kinds of pathogen reduction technologies, things like charcoal filtration and some of the other standard ones that might be out there. And we landed on E-beam because it allows us to process the material without overheating the product. And once we did that, we found that the expansion rates, both in MSCs as well as in immune cells, were really pretty good. Minor differences, of course, between the irradiated and the non-irradiated product, but we pretty much limited that. But one of the advantages we found was an improved experience using immune cells. So when we grew T cells with it, we saw that worked pretty well. And in fact, we've got quite a bit of data on some improved phenotype with the Enliven and T-Liven products. I think if you start with a non-irradiated product, it's really important, though, to evaluate what the differences are. It's not just a simple swap out. If you're just going to hit your raw material with irradiation and then hope it works, you may be in for a surprise. Again, on the manufacturing side, you've probably seen a lot of different kinds of things, and you can probably speak to that variability a little better.
Yeah, I mean, I agree, Sean. From when you switch from something that's non-irradiated to something that's irradiated, obviously, from a regulatory and a manufacturing point of view, you have to prove its compatibility and comparability. And with that, you need a substantial validation protocol and package to support that change. You want to assess the growth kinetics, the doubling times, your yields. You want to look at the cell surface characterization and most important, your potency assay at the end. How has it affected? As it says in the the ATMP regulations, it's about how the product can affect the intrinsic factors that's in these products and how it can affect your product at the end. And I suppose that's critical to assess when you do such a change. And the regulators will request that if you start with a non-irradiated product and switch to an irradiated product, they need to be sure that the comparability of the product at the end is sufficient to allow to proceed, and that you will get the same effect from that product rather than with the same as what you would have used if it was the non-irradiated version. So I think the validation of that is really important, and to investigate that.
Very good. Well, that's what we got for today. Sean, Aoife, thank you guys so much for taking the time to do this and share your presentations. For everybody who registered, we will get a copy of this webinar out by the end of this week, by Friday. And we hope everybody has a great day. Thank you so much.